So those three cancer types are the most pronounced signals felt. But in general, there was a trend towards signal across the adenocarcinomas.
I see. So there's indications that aspirin could have an effect
Yeah,
across
yeah,
at least those cancers.
yeah, and probably broadly across adenocarcinomas.
Hmm.
Now, additional studies, observational studies, have looked at whether this aspirin chemopreventive signal, so this ability to prevent recurrence, um, is associated with any underlying genetics of the primary tumor,
Hmm.
because this is the adjuvant setting. And one set of major study that showed that colorectal cancers with PIK3CA mutations, this is a gene encoding PI3 kinase alpha, which plays an important role in driving a subset of human cancers, was associated with aspirin's protective efficacy.
Hmm.
But another study also showed that HLA class 1, this is the MHC molecule that presents
Yes.
anti T cells, that molecule, if cancers didn't express very highly that molecule, then aspirin lost its protective efficacy.
Hmm. Hmm.
So
And that,
essentially, the colorectal cancer patients that best respond to aspirin and its ability to prevent recurrence in colorectal cancer appeared to be dependent on high levels of MHC expression.
yeah. And would that imply that those are people whose T cells would be the maybe better able to then recognize the cancer and do something about
Yeah,
it?
I mean, that would imply it, yes. But these are observational studies, and I think what we really now need is high quality, prospective, randomized studies and detailed biomarker analyses to really determine the immune and non-immune signals that might come into the
Yeah.
play.
And so, when people are thinking about, you know, science holistically, and we think about the connection between the preclinical and the clinical world and the mechanistic work and the translational work, what
Yeah.
you're basically saying here is you sort of filled a gap where you looked at the literature, you found some interesting stuff, you've gone in these mouse models and shown mechanistically how this story works. And the existence of this study now would be a very good justification to go in and do human clinical trials of some kind.
Human clinical trials and more detailed biomarker analyses. I think the study adds to a growing understanding of the prostanoids in general,
Yeah.
prostaglandins and thromboxanes, at almost all stages in cancer development. We know inflammation drives cancer,
Yeah.
and that in some cancer types, prostaglandin E2 driven. We know that inflammation can potentiate suppression of anti-tumor immunity in the progression phase of cancer, and that can be also related to prostaglandin E2. And here we add that actually the other prostanoid, thromboxane A2, may have this important role in suppressing T cell immunity to cancer metastasis. It's possible that prostaglandins also play a similar role in
Mhm.
cancers that might express COX2.
Yeah. And, to the extent that all of that is true, so your story is about aspirin and the metastasis
Yeah.
of cancer, but there could be more. There could be something more general going on here where
Yeah.
if you've got too much of this arachidonic acid
Mm-hmm.
pathway happening, it might
Yeah.
promote metastasis and the maybe even the origination of cancer as well, to the extent it's inflammation driven through
You're
that
very
pathway.
right. So the strongest and highest quality signal so far in humans for aspirin is in its ability not to prevent metastasis,